Okay, well, we're going to get started. Welcome. I'm Marie Harvey, Associate Dean for Research in the College of Public Health and Human Sciences, and I'm delighted to see people participating in this seminar. We are doing it both in person and via Zoom. Therefore, we're going to be using this microphone so everybody can hear. I am also very delighted that I get to introduce Karen Hooker, professor in Human Development and Family Sciences, who has graciously agreed to introduce our speaker today and to moderate this session. And she will tell you a little bit about how those of you who are on Zoom, so those on Zoom, welcome, can ask questions. Okay, so let's get started and let's welcome Karen.
Thank you, Marie. Well, welcome, Suzanne. We're so pleased that you have chosen to spend part of your sabbatical with us, and I hope you've had a chance to talk to a lot of people while you were here. So, Suzanne Segerstrom is a professor of psychology at the University of Kentucky and well known for her work on immunology, neuroimmunology. Dr. Segerstrom earned a BA in psychology and music from Lewis and Clark College and was named the 2004 Outstanding Young Alumna. After completing master's and PhD degrees in clinical psychology at UCLA, she completed her clinical internship in psychology at Vancouver Hospital and Health Sciences Center at the University of British Columbia. Dr. Segerstrom also earned an MPH in biostatistics from the University of Kentucky. She's won numerous research and mentoring awards at the University of Kentucky, and she's a fellow of 6 professional societies. Dr. Segerstrom has been awarded the prestigious Templeton Positive Psychology Prize. She's the past president of the American Psychosomatic Society, and she's the incoming editor-in-chief of Psychosomatic Medicine. Dr. Segerstrom is currently working on 2 large NIH-funded studies. One examines the health consequences of motivation and goal pursuit in older women in a longitudinal burst design. And the other is a longitudinal study of the effects of self-regulation on immunological and brain health in older adults. I'm very much looking forward to hearing her presentation today titled Well-Being, Immunosenescence, and the Brain. Okay, Suzanne.
Well, I want to thank you all for being here, virtually or otherwise, and also thank Marie and Karen and Carolyn for hosting me here for the past couple of months. It's gone so fast, and if there wasn't a husband in Kentucky waiting for me to come home, I would certainly stay longer. So today I am going to be talking about interactions between well-being, immunosenescence, and the brain. A few years ago, I was getting physical therapy, and my therapist was trying to treat one part of my body by working on a different part of my body. And when that worked, I was kind of surprised. And she said very simply, it's all connected. And my work in psychoneuroimmunology, it's just another way of saying that it's all connected. So in terms of those connections, let's try this. Nope. Error here. So I'll be talking about connections between psychological well-being, cognition, risk and protective factors, including aging, and viral infection, and how those all connect to the immune system. Now you'll see that these are lines, they are not arrows. And that's because in addition to these things talking to the immune system can talk back. So it can control viruses, that's the function it's probably best known for, but it can also talk to the brain, and it can even perhaps accelerate aging. And we'd all like to draw a direct line from well-being to immune function, and there are plenty of studies that try to do that. But today I want to emphasize intersection. And that's this wandering blue line where you see that well-being can intersect with cognition or risk and protective factors or viral immunity in its relationship to the immune system. So most of what I'm going to be talking about today is this study. It's called the Thought, Stress, and Immunity Study. Karen just referenced its most recent iteration, but it's been going on since 2001 with support from the Dana Foundation and with National Institute of Aging support since 2006. Over 300 older adults in the Lexington area have participated in the study, but at any given time, the N might run from 125 to 175. The average age of the sample is 75, with a fairly large range from closer to 60 to in the 90s. And it is generally a white and well-educated sample. That's characteristic of research volunteers in our area. So they have a high education, but a large range, and we do have some ethnic diversity in the sample. Now, every 6 months, an interviewer goes to their home. They administer some questionnaires, they administer some cognitive tests. And in addition, these participants regularly have the composition and function of the immune system measured. So we're looking for all of those intersections in this study. One of the components of the immune system that suffers with age is antibody production after vaccination. So I feel like I hardly even need this slide anymore because everybody knows so much about vaccination and antibodies now. But when you get a vaccination, the immune system produces these proteins called antibodies. And what the antibodies do is they can bind, let's say, to a virus, and that will prevent the virus from being able to enter a cell. So these antibodies can prevent infection, or they can at least reduce the severity of the infection. I feel like we all know this now. So this is a summary of studies looking at responses to influenza vaccine over a period of years. And there are 3 components to the influenza vaccine. Those are the 3 clusters that you see here. And they were comparing the group of people who were less than 60 years old to the group of people who were over 60 years old. And after vaccination, both groups had an increase in antibody. That's good because that's exactly what a vaccination is meant to do. But the older people in the yellow bars compared with the younger people in the blue bars collectively produced less antibody and therefore had less protection. Now, another way that you can look at this is the number or the percent of the group that reached what's considered a protective level of antibody. And in the younger group, it was about 60%. I'm sorry, you can't see the numbers down here. You take my word for it. This is 58, in fact. The older people, this is 35. So it ranged from about 35 to 50%. So knowing about factors that can influence the antibody response to vaccination that's an important goal. Another thing that happens to the immune system with aging is that systemic inflammation increases. And we usually measure systemic inflammation with interleukins like IL-1 and IL-6, and with a protein that's related to that inflammatory system called C-reactive protein, or CRP. Acute inflammation in response to injury or infection is a beneficial response, and you need it. It's in fact a first and mandatory step in tissue healing. But when inflammation starts to smolder all over the body on a longer term basis, then it becomes pathological. And it's associated with increases in morbidity and mortality from a number of different diseases, including pneumonia, atherosclerosis, cancer, Alzheimer's disease, frailty, osteoporosis, and other mostly diseases of aging. So in one of my earlier studies, I collaborated with an infectious disease and immunization expert, Rick Greenberg, and we looked at, in older adults, the relationship between risk and vulnerability and psychological well-being. In addition to age, there are a number of different vulnerabilities for a poor response to vaccination, One of them is sex. The female immune system tends to be more reactive than the male immune system. And so being male is a risk factor for not making enough antibody after vaccination. But being female is a risk factor for producing too much inflammation after vaccination. And other vulnerabilities include things like lower education, higher BMI, poor sleep, and lower physical activity. Now, in this study, we measured psychological health with a combination of the geriatric depression scale and the perceived stress scale. And we combined them because they both have emotional and cognitive elements of negative affect or negative ill-being, I guess you would say, on one side, and positive affect and well-being on the other side. And here are the results. On the y-axis, you have the average antibody response to vaccination over a period of 5 years. And on the y-axis, you have how much distress or ill-being the person had over that same time frame. Excuse me. And you can see there's one group that had a very good response to vaccination in terms of antibody production. People who had high BMI had lower antibody response regardless. People who had lower BMI had lower antibody response if they also had high distress over that 5-year period. Only the people who were lacking the risk factor of high BMI, that is, they had lower BMI, and they also had low distress, that combination was necessary to associate with higher antibody responses. Now, when we look at inflammation, it's a little bit different. There was one group that had a bad response, that is, that they had higher inflammation in the month after the vaccine. And those are people who were older and had high distress. So people who were closer to 60 years old had fairly low inflammation in the sample. People who were closer to 90 years old also had low inflammation if they were also not distressed over that 5-year period. It's this combination of risk and distress that was associated with higher inflammation. This is a little bit similar to something that Eli Putterman reported a couple of years later. And he was looking not at vaccine response, but at another parameter, which was telomere loss. And telomeres are these protective caps on the ends of chromosomes. You can see that they look bright in this image. And the purpose of these telomeres is to protect the DNA from degradation when the cell divides. And in fact, when telomeres get short enough, in some immune cells, they will stop dividing to prevent that outcome. So what he was looking at is stress, in this case, major life events, and healthy or unhealthy behaviors, which included diet, physical activity, and sleep. And he also found that in the presence of major life events, it's the people who were also engaging in unhealthier behavior that had the fastest telomere loss. Another element of well-being that I'm interested in is repetitive thought, which is just thinking attentively, repetitively, or frequently about yourself and your world. And there are a number of different kinds of repetitive thought. So typically maladaptive types like worry and rumination, typically adaptive types like reflection, processing, reminiscing, planning. And what we've found over several studies is that you can more parsimoniously characterize all of these different types with 3 dimensions. One dimension is valence, which goes positive, in this case on the right, to negative, in this case on the left. The second dimension that emerges we call purpose, and it has more searching, uncertain, questioning types of thoughts at one pole, which here is at the bottom, and more solving, certain, declarative kinds of thoughts, which here are at the top. And then there's another really interesting dimension, which we call total. And that just reflects the fact that there are individual differences in how much people engage in repetitive thought of all kinds. So some people think about themselves and their world a lot in a lot of different ways, and other people just don't think about that as much. Now, this total dimension gets kind of a bad rap in memes, where it's usually called overthinking or thinking too much. I have no idea where this statistic came from. Less frequently, you'll see repetitive thought presented in a positive light. So you can't really read this text down here, but it says, here's what all intelligent people do. Talk to yourself. Also think about yourself. You don't have to say it aloud. And this tweet says, not overthinking things is hard for me. I'm really good at thinking. So maybe total repetitive thought is characteristic of people who are good at thinking. Or maybe for people who are good at thinking, so-called overthinking has a different effect. So I was interested in how repetitive thought and cognitive function might intersect and relate to inflammation. Intelligence has been associated with a number of positive health outcomes, so lower likelihood of dementia, obviously, but also cardiovascular disease, stroke, and infection. And here you see that people who had a high IQ for the sample, which is a pretty high IQ for the population, it's about 1.5 standard deviations above the population mean, they had lower inflammation. People who had low IQ for this sample, but about average population IQ, had lower inflammation only if they were thinking about things frequently. They had high total repetitive thought. The only group that had higher inflammation were, I'm going to call them underthinkers. They were people with population average IQ who didn't think about things very much. Maybe they're not engaging in enough problem solving. Maybe they're not engaging in enough reflection, but also maybe they're not worrying about things that they need to be worrying about. Now, my student, Alana Gloger, who most of whom you can see here, did a pre-registered analysis of MIDAS data. And MIDAS is a large population-based sample of midlife adults in the United States. And she also was looking at this intersection between repetitive thought and cognition. She found something different. So she found that for people in MIDAS who had better cognition, if they engaged in more positive repetitive thought, then they had lower inflammation. If they engaged in more negative repetitive thought, they had higher inflammation. Now, this is interesting because we think of cognitive ability as protective, not as opening you up for risk, say, from negative well-being. So why were these two studies different? Well, one possibility is age. The MIDAS study, the range of ages was 25 to 70, so it hardly overlaps with TSI. TSI is 60 and older. In MIDAS, they assessed repetitive thought in daily diaries, whereas TSI asks about typical or trait repetitive thought. But I think the most likely explanation is that MIDAS assessed cognitive ability with a test called the B-TACT. And the B-TACT mostly involves fluid intelligence. Now, fluid intelligence or fluid reasoning is your ability to reason and solve problems independent of your prior knowledge. And it is susceptible to insult, like aging, for example, or something like maybe a brain injury. In contrast, TSI used a test called the NART, and the NART measures crystallized intelligence. Crystallized intelligence is your level of intellectual achievement, your depth and breadth of knowledge, and it is resistant to insult. So you can see in this graph from Park and Reuter-Lorenz that the fluid intelligence decreases with age, but crystallized intelligence, if anything, increases somewhat with age. And what we also found in TSI was that these two kinds of intelligence correlated differently with repetitive thought. So people with better crystallized intelligence engaged in more total repetitive thought, but not engaged in more total, but it was not related to valence. People with better fluid intelligence, in this case, a measure of executive functioning, engaged in more positive repetitive thought, but there was no relationship with total repetitive thought. So looking at these two studies together, you start to wonder if, in addition to these relationships, perhaps crystallized and fluid intelligence interact with different kinds of repetitive thought as well. Okay, let's talk about another bad thing that happens to the immune system with age. I'm sorry to be giving you a preview of all these bad things that are going to happen to you, but hopefully I'm giving you some idea of maybe how you can prevent them as well. So at the end of their life cycle, cells are called senescent, and especially terminally senescent cells. They can still do their primary function. So natural killer cells and cytotoxic T cells can still kill other cells. In fact, some of them might be especially good at doing that. But what they cannot do is they cannot create an army of clones in order to be able to effectively fight off infection. And for some immune cells, that's related to their loss of telomeres. So in collaboration with Dr. Charlie Lutz, we wanted to look at this intersection between aging and well-being and the relationship to cell senescence. This is a sample that Charlie was studying. The younger women are in their 20s. The older women are in their 70s, 80s, and 90s, all the way up to 97. And what we found is that, first of all, just as you would expect, the younger women had fewer of these terminally senescent cells than the older women. That is no surprise to anybody. But what was interesting is that there was also this very strong effect of resources, psychological, financial, social, composite of personal resources. And this effect was such that you would expect a younger woman with few resources to have almost as much self-senescence as an older woman with many resources, even though they are 50 years apart in age. One of the resources that emerged as particularly important in this study was financial resources. So these were things like adequate food and clothing, medical insurance, transportation, savings, a bigger house, all of those kinds of things. And so we kind of got interested in that in one sense, and I'm going to sort of back up a little bit and go forward. So I'm going to back up and talk about why immune cells age. So on this side, you have senescence in the T cell compartment and in the NK cell compartment. So these are people who have few senescent cells. The people up here have senescent T cells. The people over here have senescent natural killer or NK cells. And the people in this quadrant have both. And I've colored each of these dots, which represents a person by age, with purples and blues being young, old, and oranges and red being more old. And we know there's a relationship, but you can't really see it. You can't eyeball it. Now look at this. This is the same people. The blue triangle people are clearly doing pretty well in terms of self-senescence. The green dot people are clearly not doing as well. And the difference between them is that the green dots have been infected with this virus called cytomegalovirus, or CMV. CMV is a latent infection. Once you get it, you have it for life. It establishes itself in your epithelial cells, and it just stays there. And so the prevalence of CMV will increase with age as more and more people get infected, reaching about 80 to 90% in older age. There's also a socioeconomic gradient to CMV infection. So the solid line are people in the highest income quartile in the United States. The dotted line are people in the lowest income quartile. And you can see that at almost every age, people in the higher income quartile are less likely to be infected than people in the lowest income quartile. So this is interesting to us because financial resources were most important in this previous study. We know that CMV can age natural killer cells. So is that the connection? Like the older adult population in general, most people in TSI were CMV seropositive, but about a quarter were seronegative. So we could look at this intersection between well-being, age, and CMV infection. And what we found is that, again, more resources were associated with lower cell senescence, this time in the T cell compartment. And this relationship was not moderated by gender. So this applied to men and women equally. That was one of our questions since our previous study was in women only. It was not moderated by age, and it was not moderated by CMV status. But when you, let me just show you this first. There we go. In this study, what the primary driver of this were psychological resources. So feelings of well-being, hope, optimism, sense of humor, motivation, and the like. We think that this perhaps might have changed because in the previous study, data were collected in 2007, 2008. And what was going on in 2007, 2008? Great Recession, right? And so the fact that financial resources emerged as most important might have been a function of the context. So it's interesting, it's a different kind of resources that emerge here. Nonetheless, when you put CMV serostatus in the model, it accounts for about 50% of this effect. Now, how can that be? Like, how is CMV participating in this relationship between psychological resources and immunity? Well, one possibility is this. Psychological resources are also patterned on SES. So they may have some overlap, CMV infection and psychological resources. The activity of CMV in the body is also important. So this is a study led by my former postdoc, Rebecca Reed. She's now at University of Pittsburgh in the psychology department. And she was looking at, again, these are TSI data, but now she's looking at whether CMV is relatively inactive in the body or whether CMV is relatively active in the body. And what she found is that people who had high stress across the study period had higher senescence in their cells. People who had low stress, but CMV was very active in their bodies also had high senescent cells. But a combination of low stress and low CMV activity was associated with less senescence. Okay, now recall I said that the reverse direction can be true, and this is really important to remember, especially in cross-sectional studies. For example, the Midas data were cross-sectional. And so it's possible that instead of inflammation being the outcome, a combination of low inflammation and positive repetitive thought is what results in better cognition. And this intersection between well-being and risk and cognition also emerges in the Alzheimer's disease literature. So I have a 2020 publication in Journals of Gerontology that reviews the literature on personality and Alzheimer's disease risk. And what the literature suggests is that given the same amount of pathology or the same amount of genetic risk, that people who are higher in neuroticism are more likely to have clinical Alzheimer's disease. People lower in neuroticism are less likely to have clinical Alzheimer's disease, given the same brain, essentially. So that's really interesting. We have some sort of interesting data from TSI about this. What this shows is in the center, this is when the person's IL-6 was exactly their own average. On this side are times when their IL-6, and a pro-inflammatory cytokine, was higher than average. On the left is when it was lower than average. The red line is sort of the population estimate. And what it indicated is that 1 unit change in log IL-6 was the equivalent of about 1.5 years of aging in memory. So what's on the y-axis here are results from a word list learning task. So a list of 15 words is presented 5 times. After each time, you have to recall as many words as you can. So you can have up to 75 words correct. And that's near the top and the bottom. But what we're really interested in here was this slope. And what we also found, although it was not statistically significant, is that people who were higher in neuroticism, who have these paler lines, had steeper slopes than people who were lower in neuroticism with these flat lines. So this is another indicator that possibly well-being can buffer against other kinds of biological risk for poor cognition. Now, one of the reasons inflammation might be related to cognition has to do with a hormone called cortisol. And cortisol is induced by inflammation. It is anti-inflammatory, so it actually will sort of cool the fire of inflammation. But cortisol is also associated with neuronal loss, primarily in the prefrontal cortex and in the hippocampus, which are areas involved in learning and memory. So we were interested in cortisol and learning, and in collaboration with Sandra Sefton, who's a cortisol guru, and Fred Schmidt, who is a neuropsychologist, we looked at the relationship between cortisol and performance on this word list learning task. What we found is that indeed people who had high cortisol, chronically high cortisol, I should say, had poor learning. Now, an interesting thing about these word list learning tasks is that usually when you recall the words, you'll say the last words that you heard first, because they're in working memory, the kind of memory that you use to remember a phone number until you can dial it. In contrast, then you start to recall words from earlier in the list, those would come next, and those words have to make it into long-term memory for you to be able to recall them. And so this pattern where working memory is maintained, but more long-term memory declines is characteristic of people with chronically high cortisol. It's also associated with pathological memory problems like Alzheimer's disease. If there is any good news, I try to give you a little good news every once in a while. It's that temporary fluctuations in cortisol that were associated with stress did not affect memory. It was only chronically elevated. Okay, so now I want to bring infection back into the picture as we're reversing this, these lines, the direction of flow of these lines. These are data that I've analyzed and have written up in collaboration with our new neuropsychologist in the department, Justin Carr. And here we're looking at immunological aging, especially CMV infection, demographics, and well-being. In this case, problems in self-regulation. And one of the reasons that we were interested in self-regulation in particular are findings like this. These are midlife adults, and 7 of these 8 latent virus infections, including, 7 latent virus infections and some parasitic infections as well, but these are all lifelong infections. 7 of the 8 of them were associated with poorer cognition in midlife. That includes CMV and this parasite called toxoplasma. So what we found is that people who were CMV positive had poorer self-regulation than people who were seronegative. And this was in the domain of metacognition, which includes things like organization, task monitoring, and working memory. And also in behavior regulation, which includes things like emotional control, inhibition, and shifting. And even though it looks like the red line where the seropositive people is increasing over time, whereas seronegative people are remaining pretty stable, we didn't have enough power for that interaction to be statistically significant. So I should give you that caveat. Now, what about this parasite? This parasite, which is shown here, is called Toxoplasma gondii. And unlike CMV that it infects epithelial cells, this parasite takes up residence in your brain. And so it's been of interest to see what the effects of toxoplasma might be. The toxoplasma life cycle goes mainly through cats and mice. So cats have it, it comes out in their feces, mice and birds are exposed, another cat eats that mouse or that bird, and the parasite continues its life cycle. But of course, people can also be exposed to cat or other animal feces that have toxoplasma in them, and then they can get infected. And the main reason why people are like, what's the effect on human behavior of this parasite is its effect on mouse behavior. So the mice who become infected with toxoplasma lose some of their fear of cats. They don't care. And so by not really caring, they get eaten by a cat, and therefore the parasite gets to continue its life cycle. So the question is, are humans with toxoplasma also less fearful, potentially more impulsive, or poorer at self-regulation? Toxoplasmosis is like other latent infections, increasing with age. In this case, the prevalence in older adulthood is lower. It's about 40%. And that's true in TSI as well. So about 3/4 of our sample was seronegative and 1/4 was seropositive. The effects of toxoplasma on self-regulation, though, even though it lives in the brain and CMV does not, were not as clear. So for one thing, it only was related to behavior regulation. It was not related to metacognition. For another thing, there was no main effect of serostatus on self-regulation, but a number of studies have reported interactions with sex and gender. And so we looked at that as well. And lo and behold, we found that among women who were seropositive had more problems in behavior regulation. Men who were seropositive had. So I guess that's nice. The problem is that all of these interactions look different. This interaction looks different from other interactions. So for example, some studies have found that it's the men who are seropositive who have more problems and are more impulsive. So that leads them to posit that maybe it's the combination of testosterone and toxoplasma that gives people problems. But that's obviously not true here because in fact, the men who are seropositive had fewer problems. Well, maybe it's female hormones then. Well, okay, maybe, but this is a postmenopausal sample. So there's not a lot of female hormones to go around. An interesting newer theory about toxoplasma is that it interferes with the effects of folic acid in the brain, and folic acid is very important for brain health and brain function. So I'm just going to give you something to think about. In these orange bars are people with celiac disease on a gluten-free diet. The yellow bars are people without celiac disease avoiding gluten. This is a group that is increasing dramatically in the United States. It is also a group that is primarily white women, as in the TSI study. So if a gluten-free diet is low in folate, which they tend to be, it's possible that the combination of toxoplasma infection, which interferes with folic acid effects, and a low folate diet might explain why women had more problems. On the other hand, these are new data from the Nurses' Health Survey just published. They looked at gluten intake over 25 years in these women and then cognitive testing after those 25 years. And they found absolutely no relationship with psychomotor speed and attention or with learning and working memory. Okay, so that's some information, but it would be interesting to see if there were effects on cognitive change for one thing instead of one single point cognitive ability, because there are lots of things that can determine that. This is also sort of basic cognitive functions and not the sort of higher order self-regulation that you would see with impulsivity, emotion processing, self-regulation, and those kinds of constructs. So this is still an open question, but it does demonstrate this idea of intersection. How it's not just well-being in the immune system. It's well-being, it's the brain, it's cognition, it's health behaviors, it's aging, it's viral infection, it's all of them interacting with each other as a whole. Now, I'm happy to say that TSI continues. As Karen referred to, we've now added neuroimaging and measures of brain health to TSI. So our neuroimaging sub-study, of course, got shut down for COVID, like everything else in the world. But we're back up and scanning. And so hopefully in a few years, we'll be able to talk about brain health from these neuroimaging protocols in addition to all of these other factors. So with that, I want to thank my co-investigators in the TSI study, as well as all of my trainees over the years, without much of this work would not be possible. And to thank you for coming and for your attention today. So I think that leaves us lots of time for questions.
Yes, it does. Thank you so much, Suzanne, for a really stimulating talk.
Thank you.
My mind is like, I have all these questions, but ask some questions, as well as we might have some questions on Zoom. I'm guessing. Not yet, but okay.
Okay. Yes, please.
Thank you. It's fascinating.
Thank you.
Fascinating hypothesis and models. So everything's very complicated.
It is very complicated. I know we would like things to be simple. Wouldn't it be nice if it was as simple as like, look, well-being is correlated with better immune function. So if you have good well-being, you will be protected against immunological aging. But it turns out you have to have good well-being, possibly plus some other things going on. And it's that complexity that makes this work hard on one hand, but interesting on the other hand. Yeah.
It's thinking about other things. We did some work in the ARIC study many years ago and found that CMV is also related to atherosclerosis. Can be found in the endothelial cells, and we saw it related to carotid medial thickness and also with clinical cardiovascular disease, particularly among diabetics. And of course, cardiovascular disease is also atherosclerosis associated with cognitive function. So I'm wondering if you have also looked into that and see whether if you have measures of cardiovascular function and cardiovascular endothelial reactivity or other measures that could also see if that could be in part of the mechanisms or the interactions you're looking at.
Yeah, that's a really good question, because it's true that CMV, there's a direct infection of the epithelial cells. CMV infected immune cells, especially in older adults, also secrete more pro-inflammatory cytokines. So it could be a secondary effect of inflammation on the vessels. But we do have some, obviously not the kind of measures that you have, but we have sort of basic cardiovascular health. We have heart rate variability, blood pressure. So we can look at those things, and it is important. It is important to look at that intersection, even with something like dementia and Alzheimer's disease, because the presence of sort of Alzheimer's disease pathology is greatest in people who also have cerebrovascular disease. So it seems like you have to have this kind of two-hit to get clinical AD, maybe plus neuroticism. You got to have a lot of bad stuff going on at once, which is good news in a way. But you know, certainly thinking about the inflammation route, the epithelial route, the combination of cerebrovascular disease and AD pathology like A beta and tau, that's kind of the way of the future, I think. So that's a great other domain, right, that needs to come into this model, is this vascular health piece. Yeah, great question.
I think in the future we'll call it Alzheimer's diseases. In fact, I think many people are already referring to Alzheimer's disease like that.
That's right. That's right. And you know, there's a question of what is the disease? Is the disease the presence of the pathology in the brain, like you have the plaques and tangles? Do you have Alzheimer's disease, even if you're still functioning well cognitively? Or do you also have to have the cognitive dysfunction in addition to the pathology to have Alzheimer's disease? And now that we have imaging for A beta and tau, there's much more discovery of these people who have plenty of AD pathology in the brain, but cognitively they're just fine. And so I feel this kind of struggling with those people, like, do they have the disease or not? Yeah, yeah.
Did you look at anybody who had the CMV along with the toxoplasmosis?
Yeah, the cell sizes get really small for that. So I didn't try it. I think we'd need to have a much bigger sample, because you take like 0.25 times 0.75 and you get like 15%. And so it's hard to separate those people out. There wasn't a correlation between CMV and toxoplasma infection. So those two were independent of each other. We know that much, but
I don't know that much about these, and I was kind of shocked that at older ages the rates are so high. And could you tell? How would you tell?
Yeah, you don't know typically. Typically, these latent infections are asymptomatic. There are some, like some of the herpes viruses, right? You'll get a herpes outbreak at first, and then it kind of goes latent, and it might have further outbreaks. The chickenpox virus, like you get the virus, it goes latent in your neurons, and then it can come back later as shingles. But CMV, Epstein-Barr virus, toxoplasma, usually even the acute infection is asymptomatic, or you might just kind of feel like you have a cold, you're a little off for a day or two, but nothing that you could really put your finger on. And then the latent infection is almost always asymptomatic. So you have to be very severely immunosuppressed for those to cause problems. And we know that because, for example, people with AIDS get CMV blindness. CMV causes blindness in people with AIDS. So you have to be like, not just HIV positive, you have to have like very few T cells left. And then some of these latent viruses emerge and start to cause problems. But for most of us, we're carrying it around and we never know.
So this is a really stupid question, maybe, but do people with cats, are they much more likely to have toxoplasma?
Yes, they are. If it's an outdoor cat.
Oh, but not indoor.
Right. An indoor cat isn't getting exposed to the mice and the birds that have toxoplasma. An outdoor cat, first of all, is killing birds, and that's not a good thing. But it's also potentially being exposed to toxoplasma, and then it's being toxoplasma back to you. And then you have these parasites in your brain, which is just a creepy thing to think about. Dog people. Yes, we dog people, we don't have that problem. So, there you go.
Are there any Zoom questions?
Not yet.
Okay.
I can shift gears a little.
Yes, please.
This is all. This is really, really fascinating. Thank you. And
Thank you.
I'm sort of sticking on a thing that I know a little bit about at least, which is around these psychological resources and these other pieces. And I'm just wondering from like an emotion regulation piece, you're talking about the positive and negative, and that have you also looked at things like ways in which people not necessarily cope, but strategies that they have, so whether it be like you use your social support network, or you ask for help, or you just like distract yourself or exercise, like do you, and again, knowing nothing about like other pieces, like are those also potential protective mechanisms that people would have learned?
Yes, yes. And in fact, Rebecca Reed is working on that. She did, I think her paper, she has 2 papers, one using MIDAS data and one using the TSI data. The MIDAS data paper, I think, is out. And she's working on the TSI paper. She really carved out that emotion regulation piece for herself. The resources piece comes from conservation of resources theory, which I see a couple of people nodding, but it's like one of my favorite theories because it's not just like a stress theory. It's about both sides of the coin. It's about resource gain, how that happens, how resources in one area help another area. So instead of just looking at social support, you're looking at this big picture of like, well, maybe your social support also helps you psychologically, also helps you financially, maybe helps you gain status. So they're feeding into each other. They're upward and downward spirals. It's a great theory. I really love it. So if you haven't read Hobfall, conservation of resources theory, it's a theory that I think does not get the traction that it deserves in health psychology. Yeah. Carolyn, sorry.
So no, I have lots of questions, and I wish we had more time. So one of the things I'm wrestling with is that we've been looking at longitudinal changes in stress, and we find that people with elevated life events are more likely to die, which is obvious reasons. But we also found that people who rated their hassles as more intense are also more likely to die. And the 2 effects were bloody independent. So I'm trying to figure out what are possible pathways through which these related different types of stressors might be having independent effects on mortality. And that was one of the reasons for the paper that I showed you that I had been working on. Looking at that. I'm just curious if you have any ideas.
In a sort of global general sense, one thing that I would wonder about is whether they have different intersections with other risk and protective factors. So you think about Eli Putterman's data where life events accelerated telomere shortening, but only among people with unhealthy behavior. If you think about hassles might intersect with something else, personality, and have some different outcomes. So when you're looking at something as global as mortality, there are lots of ways to get there, and we're all going to take one eventually.
Right. And neuroticism, by the way, was in the equation, and it had independent effects.
And it had independent effects as well. So yeah, I think there are all these pathways, and depending on what intersection you might have, right? So you might have, and if you look at Eli's data, hopefully, here we go. Let me see if I can get back there. No, I'm finding it. There it is. If you just looked at life events, there would be a main effect of life events on telomere shortening. But if you didn't look at the interaction with healthy behavior, you wouldn't realize that the people who are really getting the short telomeres are the people who have life events and not like a mediation, and this interaction with unhealthy behavior. And so that's what I think. I wonder if life events interact with healthy behavior and daily hassles interact with something else. And that's how they can have independent effects of each other, is because the life events are accelerating the health behavior pathway, and maybe the hassles are accelerating some other pathway.
I wonder, I mean, I just off the top of my head, variability in blood pressure or
Mhm.
heart rate function or something like that.
Yeah, it might be that it's this idea of allostatic load, that, you know, these hassles that are sort of constantly coming and going are increasing the allostatic load. Maybe that's the pathway by which hassles are leading to mortality. So yeah, it's very hard to say when you have, you know, what has been called the ultimate outcome in health psychology, which is mortality, or premature mortality risk, we should say. And there's so many ways to get there. Yeah.
I've got time for one more quick question.
Okay. Any in the room? Any on chat?
And if not, thank you again for a very.
Thank you. Thank you very much. Thank you.